Okay, welcome to our AP Biology Unit 5 exam review. Sorry again I can't be there and that there's no pizza, but hopefully this will help you study for the exam coming up. So we are going to play Kahoot. It's going to be 1 hour long. You're going to want to make sure that you're watching this video at the same time as playing the game. This video is pre-recorded, so you will only see me playing, and I'll do a terrible job so that we can keep the timing on track. Now, as you join the Kahoot, I want you to use your last name and first initial so I know who is playing. Any students who play the Kahoot for the entire hour will get 5 points extra credit added to their Unit 5 exam. So that's a pretty big benefit for those of you that are playing. So again, as you join the game, we're playing from 4 p.m. until 5 p.m. Make sure that your name on the game is your last name and first initial. I'm going to let this sit for about 4 minutes until we get everybody added from the class. Okay, about 2 more minutes to get joined into the game. If you don't get joined during this initial time, it will show the game pin throughout the game so you can get added again. But again, you do need to play for the entire hour to get the extra credit. You will be able to go back and watch this video at any time before the exam to review for the exam. Remember, you should be adding yourself as your last name and first initial only. Please don't put your other funny names. I won't know who you are if you do that. About 1 minute remaining. So it's currently 4:03. We will start in 1 minute. Make sure that you are playing live on your phone with the Kahoot app and that you are watching the video while we go. Otherwise, you won't see the correct questions match up to the answers that you are given on the app. About 30 seconds remaining and we will begin. All right, let's go ahead and get started on our game. So question number 1, Mendel called those traits that were not expressed in the F1 generation recessive. Good job to those of you that got it right. Expression of a trait such as yellow versus green seeds are referred to as These are phenotypes. So phenotypes are the expression of traits. Alternate forms of the same gene are known as alleles. For those of you that are still playing with me, you may want to get a piece of paper handy. Some of the questions will have longer time because they will be on probability and Punnett squares. Let's go to the next question. The information in the squares within a Punnett square are offspring genotypes. So although we may know the phenotypes based on the genotypes, it only tells us the offspring genotypes. A red-eyed heterozygous female Drosophila is mated with a red-eyed male. What are the offspring ratio? Now you should know something about eye color and Drosophila. These are fruit flies for those of you that don't remember. You've got quite a bit of time to figure this out. Today we're not working with who is the fastest. Today we are trying to get answers correct. So go ahead and work through the problem. The answer is all of the females will be red-eyed, half of the males will be red-eyed, and half of the males will be white-eyed, and this is because eye color in Drosophila is a sex-linked trait. When a trait such as skin color in humans is continuously variable over a wide range, it is considered So this is polygenic inheritance. Now remember, early on humans thought that there was this blending hypothesis, but things like skin color and human height show polygenic inheritance. This means there are multiple genes coding for the production of a trait. Genes that occur on the same chromosome are said to be So these are linked genes, genes that are linked. They exist on a same chromosome. What is the difference between a monohybrid cross and a dihybrid cross? Dihybrid crosses are crosses between two heterozygote genes, while monohybrid crosses are between one heterozygote gene. So we're looking at one gene where both of the individuals are heterozygotes with a monohybrid. If you hear my puppy howling in the background, she's upset. A cross between homozygous purple flowers and homozygous white flowers results in all purple flowers. What kind of inheritance is shown? So this is the completely dominant kind of characteristic between these purple flowers and the white flowers. All right, next question, number 10. How many unique gametes can be produced from an individual with the genotype shown? So you have an entire minute to figure this out. We're looking at how many unique gametes can be produced. About 5 seconds left. So the answer is 8 unique gametes can be produced. Next question, number 11. 2 plants are crossed, resulting in the offspring with a 3-to-1 ratio for a trait. This would suggest which of the following? So the answer is that both of the parents were heterozygotes for a trait. We saw this in Mendel's results between his F2 generation and the F3 generation. 2 characters that appear in a 9-to-3, 3-to-1 ratio in the F2 generation should have which property? Read through these very carefully. All right, so these genes are obviously obeying the law of independent assortment. Now this 9-to-3 to 3-to-1 ratio is what Mendel found when doing a dihybrid cross. So these would be our expected results. Now what we find sometimes when doing a dihybrid cross is we do not get these results, and this helps us determine whether genes are linked or unlinked. Question number 13. An animal with the genotype shown could produce which of the following gametes? This should be an easy question for you guys. So we're looking at gamete production from a genotype that is given. So the answer, of course, is uppercase H, uppercase T. You cannot have a gamete that contains 2 alleles for a single gene. So hopefully that was a real easy one for you guys. All right, question 14. If the uppercase R leads to red and the lowercase r leads to white, and you're given a cross, what is the chance of these individuals having a white colored offspring? These exhibit complete dominance, by the way. So there's a 50% chance. Now, while using Kahoot, I'm pretty restricted on how many characters I can use when typing this out. So I had to get a little bit clever with my wording. All right, next question, number 15. Which of the following is false about the law of segregation? You have an entire minute to figure this one out. Please read them carefully. About 10 seconds left. So the law of segregation talks about all of these things, except it is obviously not used to determine the number of chromosomes within a cell. So I'm doing terrible at this, but that's on purpose so we can keep our timing correct. Let's move on. Question 16. The fact that all 7 of Mendel's pea plant traits obeyed the law of independent assortment would tell you So this would mean that Mendel was super lucky in the traits that he picked and that all of the genes for his traits were on different chromosomes. All right, question 17. Mendel's observations of the law of segregation in gamete formation is based on... All right, so this would be anaphase 1 of meiosis. The law of segregation is discussing the separation of alleles, and so we're looking at when those homologous chromosomes are separated from each other. All right, next question. Mendel's law of independent assortment has its basis in which of these events of meiosis? So this would be shown during metaphase 1 of meiosis when those tetrads align at the equator. This is saying that different genes will be independently assorted from each other. All right, question 19. We're going to look at a picture. The wording was too long to get into it. So black fur in mice is dominant to brown fur, and short tails are dominant to long tails. What fraction of the offspring from the cross given will have black fur and long tails? 5 seconds left. The answer is 1/2. All right, tall is dominant to short. A heterozygote crossed with a homozygous tall will have what percent of their offspring be short? So this one should have been super easy. If you have an individual that is homozygous dominant for a trait that shows complete dominance, they cannot have an individual that shows the recessive trait. So 0% of their offspring will be short. All right, again, we're going to look at the picture. So this one's talking about true breeding pea plants. They give you a lot of information. I clearly should have left more time on this. You only have 8 seconds. I want you to know how to do this one in real life, but you may not have time to calculate this right now. So the answer is about 190. We'll do that one in class right before the exam. In a cross given between the two genotypes, what is the probability of producing an offspring that is completely dominant, so homozygous dominant, for each of the traits? So it is 1/64. Now, hopefully you did not create a Punnett square with 64 squares because you did not have time for that. So you should have used the law of multiplication to be able to figure this out. If you don't know how to do that, go back to the Prezzies and figure out how to do that. All right, we're going to use the picture. And what we want to know is between the information given and our answers that we have, we want to know what kind of relationship is going on between these genes. All right, so this is epistasis. This is when one gene affects the role of another gene. So we're looking at one gene affecting how another gene behaves. Okay, again, we're going to use the picture. We want to know how many genes are responsible for coat color in Labrador retrievers. This is the same picture as before. So how many genes are responsible for coat color in retrievers? So there are 2 genes responsible for coat color in retrievers. All right, based on the picture, what kind of inheritance is discussed? So this is codominance. If we see both of the characteristics show up in the offspring, then it means it is codominant. This is a codominant trait. I'm still doing terrible, but we're getting there. We're about 25 minutes in. We're going to use the picture, and we want to know what are expected of the F2 generation. So F2 phenotypic ratios. We are given the F1 generation, so you are going to have to do some calculating here. So the answer is about half of them are going to have mixed prongs. That's that codominance. A quarter of them are going to be two-pronged, and a quarter of them are going to be one-pronged. What are the possible blood types of a child from a man with type A blood and a woman with type O? Now remember, I'm not giving you their genotype, I am only giving you their phenotype. So what are any of the possible blood types of the children? So the answer is their children could have type A or type O. Okay, question 28. What are the possible blood types from a child with a man that has type AB and a woman with type O? So the answer is they can have children that have type A blood or type B blood. They cannot produce children with type O, and they cannot produce children with type AB. All right, next question, 29. Which best describes when phenotypes of a heterozygote differ from the phenotypes of homozygotes? So this is incomplete dominance. We talked about this in class, right? I'm not always going to give you red flower and white flower equals pink flowers. Sometimes we'll talk about blue chickens. So remember, when that phenotype of the heterozygote does not look like the phenotypes of either of the parents or those homozygotes, this has to be incomplete dominance. Question 30. So we're going to use a picture to answer the question. So the answer is 50%. Since this is a dominant trait, that parent is a heterozygote for this trait, so they have a 50% chance of their child having that condition. A woman has 6 sons. She's crazy. The chances that her next child will be a daughter is what? So the answer is 50% chance that it will be a daughter. Remember, past events do not change the probability of current events. So each time a person has a child, there is a 50% chance it will be a son and a 50% chance it will be a daughter. What is the genotype of the individual, generation 2, number 5? So we're looking at a pedigree here. And we're given some genotypes in the very first generation. Now you should be able to figure out the genotype of any individual based on the information given. So this individual has to be homozygous recessive. Next question, number 33. What is the probability that individual generation 3, number 1, is heterozygous? So there is a 100% chance that that individual is heterozygous, and that's because that individual's mother is homozygous recessive. What is the chance that a couple will have 4 offspring, all are female? So now we are looking at 4 offspring in a row that are all female. Hopefully this was easy. We talked about this in class. So there's a 1/16th chance of having all 4 of your offspring be one of the sexes. And this is now we have to look at the probability of each event and then add those together. A hemophiliac male and a normal female have a hemophiliac daughter, which is a false statement. We're looking for a false statement. So the one that is a false statement is that the daughter inherited a dominant allele from her mother. So hemophilia is a sex-linked trait. Hemophilia is recessive, however, it shows up in males quite often, and this is because they only have one X chromosome. So all of those other statements are correct about hemophilia and this situation. That mother has to be a carrier of hemophilia. The daughter had to have inherited a recessive gene from each of the parents, and of course the genotype of the father is XY, with that hemophiliac gene being contained on the X chromosome. Which of the following reveals the central dogma of biology? So DNA is transcribed into RNA, which is translated into a protein. During transcription, blank binds to the blank region upstream of the blank. So the answer is that during transcription, RNA polymerase binds to a promoter region that is upstream of the gene. In eukaryotes, transcription requires all of the following except So in eukaryotes, we require transcription factors, RNA polymerase, and a promoter region, which is usually a Tata box. We do not require an operator, and that's because operons and operators are found in prokaryotes only. If coding DNA strand reads 5' ACCGTA, then 3', the corresponding mRNA would read, be careful with this, read the question carefully. So hopefully you immediately saw the answers that had T's in it and knew that it couldn't be those ones. Now, a lot of you will have missed that, and that's because this was a coding strand of DNA, not the template strand. RNA polymerase reads a template strand, which would be complementary to what we see above. So the template strand would have read 3' T, G, G, C, A, T, 5'. So the answer of the mRNA looks a lot like the coding strand, and that's why we call it the coding strand. Which of the following RNA molecules is incorrectly matched with its function? So the one that is incorrectly matched is tRNA, and that it carries a copy of the gene sequence. Remember, tRNA brings an amino acid to the ribosome. Now, the answer that is in green, miRNA, that should have said siRNA. So if you selected that, good for you. I have that incorrectly matched. But hopefully you recognize that the tRNA was also incorrect. Which of the following must happen before translation can occur in a eukaryotic cell? So all of these things have to happen before that pre-mRNA can be removed from the nucleus to get into the cytoplasm. All right, question number 42. A single nucleotide is deleted from DNA, which is considered what type of mutation? So this is a frameshift mutation. So any kind of nucleotide deletion or insertion causes a frameshift, which causes a shift in the reading frame. So you end up getting different amino acids for every sequence after that insertion or deletion. All right, next question, 43. A nucleotide substitution causes a change in a codon resulting in a different amino acid. What is the type of mutation we're looking at? So any of these substitutions can cause any number of mutations. If we change an amino acid, so we swap one amino acid out for another, this is a missense mutation. Sickle cell anemia is caused by a missense mutation. It is a single substitution in a nucleotide. A single nucleotide substitution causes a change that introduces a premature stop codon. So this is called a nonsense mutation, and that's because when the gene in question is transcribed and the mRNA received at the ribosome starts to build a polypeptide, that polypeptide ends up being very, very short and usually is useless in the cell because a stop codon was introduced prematurely. Nucleotides in bacterial DNA near the start of an operon where the repressor can attach. What do we call this location? So this is the operator. The operator is that sequence of nucleotides where a repressor can attach, the operator can be switched on, and the operator can be switched off. A molecule that binds to a repressor and changes its shape, allowing it to switch an operon off. So this is a co-repressor. Now a lot of you might have selected repressor. Of course the repressor does bind to the operon or the operator of the operon, but a co-repressor is the molecule that binds to a repressor. So make sure you're reading the questions carefully. A molecule that binds to a repressor, causing it to be unable to bind to the operator. This switches the operon on. So this is an inducer. An inducer binds to the repressor. It causes a change in shape of that repressor, and then that operon is switched on. Now hopefully you guys remember which of the operons is inducible and which of the operons is repressible. I do not have that in this review, but they will be on the exam. Which of the following increases gene expression in eukaryotes? All right, so the answer is histone acetylation. Both methylation, which causes heterochromatin, remember that is DNA that is not red, it is not coding, and DNA supercoiling, which is that tightening up of the DNA, all of those things cause restrictions in gene expression. Only acetylation opens up the DNA to allow for transcription to occur. All right, question 49. Inheritance of traits transmitted by mechanisms not involving nucleotide sequences. Epigenetics. So epigenetics is talking about methylation and acetylation. Last question. Which of the following are ways that eukaryotes control gene expression? The answer is all of these are ways that eukaryotes control gene expression. We methylate and acetylate our genes. We have alternative intron and exon splicing. We have proteasomes, remember, which are enzymes that cut up RNA in the cytoplasm. So eukaryotes have a variety of ways to control gene expression. You'll need to be familiar with each of these. So hopefully you guys did a wonderful job on this. This is not the only information that will be contained on the exam, but go ahead and watch this a few times so you're familiar with these questions. I will have questions that are similar to these on the exam. Good luck. Have a great weekend.